To analyze expression data in GenePattern, you must first format your expression data as a GCT file or res file. The main differences between res and GCT file formats are the res file format 1 contains labels for each gene's absent A versus present P calls as generated by Affymetrix GeneChip software, and 2 does not allow missing expression values. Therefore, for this tutorial, we will use the GCT file as our template. The GCT file format is a simple tab-delimited text file containing a matrix of expression values organized as follows. The first line contains the version string, which is always #1.2 as shown here. The second line indicates the dimensions of the matrix in the rest of the file. The first number indicates the number of rows, i.e. probe sets in the file, not including the first 3 rows. The second number indicates the number of columns or samples in the file. Note that this does not include the name and description columns. Our example file therefore has 12,564 rows and 48 columns. The third line contains the name and description columns followed by the sample IDs. The rest of the file contains expression data for each of the genes. There is 1 line for each gene and 1 column for each of the samples. As previously indicated, the first 2 columns of each row will contain a name and description for each of the genes. Names and descriptions may contain spaces since fields are separated by tabs, though each ID in the name column must still be unique. Gene expression data from any source can be converted into the GCT format using one of several GenePattern modules for file format conversion or other file conversion methods. We will begin with the GenePattern modules. The Expression File Creator module creates an expression dataset from a set of individual Affymetrix cell files. For this tutorial, we will use a .zip archive of cell files as input and leave all module parameters at their default settings. For more information about the module, click the Help button in the upper right corner. The module outputs a .gct file, which can then be used in other GenePattern modules. GeoImporter creates a GCT file from a GeoSeries record or a Geo dataset. Again, more information about the module can be found by clicking on Help in the upper right corner. Here we've chosen a GeoSeries record and will leave the remaining parameters at their defaults. The module outputs a GCT file, which, as with all modules that output a .gct or .res file, can be used in any GenePattern module which accepts that file type as input. Other GenePattern file format conversion modules are CArray Import Viewer, which exports experiment data from the CArray database and writes it to a GenePattern GCT file, and the MageML Import Viewer imports data in MageML format into GenePattern and converts it into the GCT format. For some data types, a manual conversion method must be employed using a text editor or spreadsheet editor like Excel. As an example, we will briefly walk through the conversion of a CDT file to a GCT file in Excel. Delete the CLID and GWeight columns, as the GCT file format only allows for 2 columns of annotations, typically name and description. Since the GCT file format also allows for only 1 row of identifiers, we'll delete the AID row as well. Add 2 header rows at the top of the file. In the first cell of the first row, enter #1.2. In the second row, first cell, enter the number of data rows, 1553, and in the second cell, enter the number of columns, 44. cDNA files can also be manually converted to GCT in the same manner. For all manually modified files, be sure to save as tab-delimited and to use .gct as the file extension. In Excel, you may need to specify the full file name in quotations to prevent Excel from appending .txt. Also be aware that auto-formatting can change your data entries. Be sure that your data is being displayed as intended before saving. Most GenePattern modules require that all cells in a GCT file contain data. If your original file was missing data, use the Import Missing Values module in GenePattern to add the missing data to your GCT file. For further information about GenePattern file formats and modules for the file format conversion, please visit the GenePattern website at genepattern.org and the CA BIG Molecular Analysis Tools Knowledge Center.